Hello everyone, this is Kuvutoli Sumi, and today I'll be giving a short presentation on an important bacterial disease of rice called bacterial leaf blight. Bacterial leaf blight is a very common and widespread disease of rice in all rice-growing countries of Asia. It is also one of the oldest known diseases and was first reported and noticed by the farmers of the Fukuoka area, Kyushu, Japan, as early as in the year 1884. In India, this particular disease was first reported in 1951 from Bombay. This disease is characterized by two symptoms, that is seedling wilt or crescent phase and the leaf blight phase. The seedling wilt or the crescent phase of this disease is considered as the most destructive manifestation of the disease and is observed in seedlings of 1 to 3 weeks after transplanting. Initial symptoms include the appearance of green water-soaked lesions along the cut portions or the tips of the leaves. With the disease advancement, the leaves start to wilt, roll up, droop, and appears grayish-green to yellow in color. In severe cases, the entire plant wilt completely, thus killing the plant, resulting in partial or total crop failure as observed in the pictures. The other symptom is the leaf blight phase, where water-soaked yellowish stripes appears initially on the leaf blades. These symptoms may also start appearing from the leaf tips with a wavy margin. With disease advancement, the leaves undergo necrosis, turning yellowish-white or golden yellow in color, leaving the midrib intact as seen in the pictures. In humid areas on the surface of young lesions, milky or yellowish, opaque and turbid dewdrop like bacterial ooze are observed during the early morning hours. These dry up to form small yellowish spherical beads and helps in dissemination of the disease. Severely infected leaves dry up quickly and infected plants yield fewer and lighter grains, resulting in crop yield losses of up to 60%. Let's discuss about the pathogen. The causal organism of this disease is a bacterium called Xanthomonas oryzae petovar oryzae. The bacteria is a rod shape, measuring 1.2 micrometer in length and 0.3 to 0.5 micrometer in weight. They are found as single cells, occasionally in pairs, but not in chains. They are gram-negative, non-spore-forming, devoid of capsules, and forms pale yellow circular smooth colonies with an entire margin when grown artificially on nutrient agar media in laboratories. Some of the factors that favors the development and incidence of this disease are warm temperature of 25 to 30 degrees Celsius, high humidity of more than 80%, and continuous rain, strong winds capable of causing wounds on the plant, excess of nutrients like nitrogen, and deficiency of nutrients like potassium and phosphorus with closer plant spacing are also known to favor this disease. Presence of stubbles, crop debris, and ratoons of the infected plants serves as a source of primary inoculum in the field. Irrigation water and rain splashes helps in the spreading of the disease through bacterial ooze. The management strategies can be grouped under three categories, that is preventive, chemical, and cultural method. Under preventive method, we have seed treatment. Seed treatment with bleaching powder at the rate of 100 grams per liter of water and zinc sulfate at 2% per kg of seeds, or soaking the seeds for 8 hours in a solution of agromycin 0.025% and wettable sericin 0.05%, followed by hot water treatment for 30 minutes at 52 to 54 degrees Celsius can be adopted. Seed treatment can also be done by soaking the seeds in sericin 0.1% solution for 8 hours before treating with streptocycline and antibiotic at 3 grams per liter of water. 2 sprayings of neem oil at 3% or neem seed kernel extract at 5% or cow dung at 20 grams per liter of water is also recommended for the control of this disease. The first spray can be given at the time of initial appearance of the disease and another at fortnightly interval. Cultural methods of this disease management includes growing of tolerant varieties like IR20, IR72, and resistant varieties like Ajaya. Procurement and sowing of disease-free seeds is a very important cultural methods of disease management. Grow nurseries preferably in isolated upland conditions. Avoid clipping of seedlings during transplanting. Balance fertilizer application and avoiding excessive nitrogen application, which can be skipped at the budding stage of the crop if the disease incidence is moderate. Alternate wetting and draining of the field is also recommended. Destruction of infected crop residues or straws and removal of alternate and collateral with host. avoidance of flow of water from affected fields to healthy fields, adoption of proper plant spacing. Chemical methods of this disease management include spraying a combination of streptomycin sulfate and tetracycline at 300 grams with copper oxychloride at 1.25 kg per hectare. If needed, a second spray can be given after 15 days from the first spray. Application of bleaching powder at 5 kg per hectare in irrigation water is also recommended for the control of the crescent phase of this disease. Foliar spray with copper fungicides alternatively with streptocycline at the rate of 250 parts per million checks the secondary spread of the disease. These are some of the management strategies for the control of bacterial leaf blight disease of rice. That is all for the presentation. Thank you.